We'll just pull the slides up here. I talked here last year. How many people saw that talk? Yeah, quite a bit. So this talk, I'm going to cover some of the same material, but I'm going to go a lot faster through it because I want to get to the new material. We've got a lot of exciting stuff going on, and it's hopefully stuff you can use immediately for your practice to help people get better. I'm going to talk about oxaloacetate supplementation.
That's your pointer, forward, back.
Okay, and we're rolling. Thank you, Frank. Appreciate the opportunity to come and talk to you. First, I'm going to talk about calorie restriction. Now, how does that tie to everything? When I started looking at aging, the gold standard for slowing aging has been, for the last 70 years, calorie restriction. I tried calorie restriction personally because it has so many benefits, and I found it both difficult and annoying. And what was worse is other people found me annoying. My wife was so glad when I got off of it. So what we started doing at UCSD in San Diego was looking at the molecular pathways of calorie restriction. Why does it increase lifespan throughout the animal kingdom? I mean, there's almost nothing we know that uniformly increases lifespan. This is what we're looking for. But we're also looking for health span. And calorie restriction has a lot of benefits. So we're going to talk about that really briefly because I know you know about calorie restriction. So first we're going to talk about the mechanism of action here. We're going to look at some applications, some clinical trials that we're now involved in, and some exciting stuff, and then patient profiles that may benefit. So some of this I'm going to go through pretty quick. And if you have any questions, you can get a hold of me at acash@benagene, benagene.org. So we've published some of this information about extending lifespan in the animals, in aging cell, longevity science, anti-aging therapeutics. Some of the benefits, of course, the increase in lifespan. That's pretty huge. I mean, how many antioxidants do you know of that increase lifespan? None. Okay, decreases in cancer incidence in animal models. So we take human cancer tissues, we put them in animal models, we see how it spreads. 55% reduction in the incidence of cancer and much better prognosis with calorie restriction. Decreases in animal models of any kind of neurodegenerative disease like Parkinson's or Alzheimer's. Complete protection against type 2 diabetes in primates. Again, these are all animal studies because it's hard to lock people up into a room or a cage for 80 some odd years and feed them less than they want to eat. But we do it routinely with animals. We're nice guys, really. We're also looking at reduction in inflammatory diseases such as autoimmune type diseases. And even though you might think what we do to the animals is somewhat cruel, it is translating to life-saving techniques for human beings. And that would be the only reason we could justify doing that. So moving along, what we've seen in calorie restriction, and this is based on some work from MIT and Harvard back around 2004 to 2006, is in calorie restriction we see an increase in the NAD to NADH ratio. So NAD goes up, NAD goes down. And when that happens, we see nothing. There we go. We see increases in lifespan. We see increases on a human cell lifespan. We see, I mean, paper after paper after paper, increases in mitochondrial NAD lead to cell survival. And here are some newer papers. When NAD to NADH goes down, sarcopenia starts. How many of you have patients that are elderly that are starting to go through muscle wasting? NAD to NADH ratio has dropped, okay? When NAD to NADH ratio goes up, mitochondrial density grows up. We've actually been able to measure an increase in mitochondrial density in the brain. These are the power points of your cell, not the power points, but the power plants. How important is that? That's basic cellular function. If you keep the cell alive with energy, it can actually do its job. So we've also seen, and this is a, I'm trying to figure out the pointer, but I'm just not clever enough. This is a really recent paper showing that as NAD levels drop, the communication between the nucleus and the mitochondria breaks. And the nucleus keeps pushing out proteins that deal with oxidative phosphorylation, and the mitochondria that's supposed to do the same thing so that oxidative phosphorylation can continue to work, it stops producing those proteins. And so what you have is a cell where NAD levels drop, oxidative phosphorylation stops, the cell tries to survive through other processes like fermentation in the cytosol and wherever it can, and that, I think I've pretty accurately described the Warburg effect. They've now found a mechanism by which the Warburg effect is enacted. And this is work that's, if you look at this paper, there's like 100 different doctors on this. I mean, it's a multi-university, multi-faculty, all-star study. It's pretty incredible that we've found a physiological reason for the Warburg effect, but it gets better than that. When they increased precursors to NAD, they were able to reverse the effect. They were able to turn oxidative phosphorylation back on by increasing that communication between the DNA compartment, the nucleus of the cell, and the mitochondrion, so that you could get back to normal usage of oxygen. Now, is that interesting to most of you? I would think so, yes. And then also recently, they showed that as the NAD-NADH ratio goes up, breast cancer metastasis in stage 4 breast cancer goes down. And that was work out of the Scripps Institute just this last year. So this is, NAD-NADH ratio is probably one of the hottest topics around. You're going to see a lot more papers on this. Every month there's something new that just blows me away. So how do we increase this ratio? Well, the obvious one is calorie restriction. That's how we figured out what was happening, what was the beneficial effect here, is getting that NAD to NADH ratio up. Another beneficial effect is through exercise, through gluconeogenesis. You can run through the charts and you'll see, yeah, NADH goes into NAD and yeah, there's an increase. So diet and exercise. Okay, my talk's over. You know, I mean, if we could all calorie restrict and exercise as much as Frank would like us to exercise, to the point of exhaustion, to the point where you're falling down the stairs, we probably would have a heck of a lot less problems. Okay, well, we looked at another way to increase that NAD to NADH ratio. It's kind of a trick. We call it the have your cake and eat it too theory. And what we're doing is we're taking a human metabolic product, part of the citric acid cycle, oxaloacetate, and we're using it as a supplement to create malate, and through the enzyme malate dehydrogenase, which is everywhere, this occurs in the cytosol, And as part of that, NADH turns into NAD. So you're both lowering NADH and increasing NAD, and this raises the ratio. Now, you don't have to take my word for that. There was another guy who, I'm not a physician, I'm a physicist, but I've even heard this guy. His name was Krebs, and he actually measured this, and he saw that the ratio increased by 900% in about 2 minutes. Is that powerful? Is that powerful? And of course, yeah, you make some extra malate, but that's like eating an apple a day, right? So this is what we've found is going on, and we've published this. The NADH levels drop, NAD levels drop, and any time that ratio goes up, you activate something called AMP protein activated kinase, or AMPK. And this you can think of as the fulcrum of a seesaw, where it's monitoring this NAD to NADH ratio. And when it's one way, it acts with transcription factors. In this case, we saw it was the DAF16 transcription factors in worms, which is where this paper came from. But with transcription factors to open up the DNA coil to specific genes, these genes are then expressed, and you have the proteins from the genes released into the cell to do the work. Now, when the ratio's the other way, it uses other transcription factors and turns on different genes. And the difference is about 10% of your entire genome one way or the other, which is pretty astounding. I mean, here I'm not talking about drugs, or I'm not talking about strange chemicals. I'm talking about eating less. Eating less changes the output of your genes by 10% or more in different genes, turning on and turning off. We used to think that it was all about genetics. And then there was another camp that it was all about environment. Remember, I don't know if you guys remember Trading Places, they bet $1. But it turns out it's both. And AMPK is one of the master switches of metabolism that recognizes the redox of the cell. So that's what we're doing here. And when we disable either AMPK or disable DAF16, we stop seeing this lifespan extension in the animals. So we're pretty sure of this pathway. So here's the gene picture. We did genomic testing, and in the mice that we did this with, we saw about 1,700 genes change out of about 20,000. In the calorie-restricted group, less with just oxaloacetate added to their feed, and there was an overlap of 363 genes changed in common, and of these that were common, there was a 98% positive overlap. So when some genes were going up in activity, they were the same, and some genes were going down in activity, they were the same. Now, this isn't 100%. Is it good enough? Well, the way to find that out is to test it. So that's what we did. And here's just some examples of some of these genes. The FORCAD3A, which has been associated with increased lifespan, 100% increase with calorie restriction, 70% with oxaloacetate. The FOXO1A, 30%, 40%. You can see, and it goes through this throughout the 300 and some odd genes. So how can we use this? Well, first, oxaloacetate is a citric acid cycle metabolite. It's in every cell of your body. This is not a Frankenstein model. And you can see when we add it to the feed of any kind of animal, we're seeing about a 25% increase in lifespan. This is with worms, mice. This is stuff that's done by Steve Spindler. He showed an increase in lifespan all until the last 3 mice, and we're still trying to figure out what happened there. We think the oxaloacetate degraded. So this is in worms, flies, mice, increase in lifespan. In humans, they did a clinical trial showing a 25% reduction in fasting glucose levels in type 1 and type 2 diabetes. In normal people, this is fasting glucose levels, and we're seeing, again, the glucose all over the chart. After about 4 to 6 weeks, the levels drop and become extremely stable. And that's important, that drop in amplitude, because the problems occur at the high end and the low end. So this gets rid of some of those problems. And we came up with some commercial products that you've seen here. I've had a booth here, Benagine, Dr. Mitch's rock solid glucose. And here's an example of a study we did outside the US, a European woman, and you can see her levels of fasting, postprandial, and before bedtime, they all drop. Also, look at her swing at the start of the study, and look at how tight her glucose regulation is. This is because we upregulate that FOXO3A gene, which deals with glucose homeostasis. So, huge improvements. We also see improvements in fat mass. We saw about a 30% reduction in, this is using nuclear magnetic resonance to measure, 30% reduction in our animals in fat mass, but no change in muscle mass or free fluids, so that's nice. And also, after we starved them, we looked at how they gained fat, and the oxaloacetate mice rebound slower because we downregulate the genes that create and store fat by about 30%. So this, again, leads to commercial products, lean gene, but these are all the same thing. Every bottle you see in this presentation are all going to be 100 milligrams of oxaloacetate with 150 milligrams of vitamin C. Here's some other tissue protection. It's been shown to protect mitochondrial DNA in the brain. That could be important. Shown to help with macular degeneration, pancreatic islet cells. It's an antioxidant. And it's been able to increase endurance on a cellular level. We actually mounted a muscle cell on transducer plates and saw a 10% increase in endurance. The athletes that I talked to say that it gives them about a 2-hour boost, and then they have to take another tablet. They typically take one about 15 minutes before they compete. Some of these people are national medal winner athletes. We've also seen a reduction in essential tremor. It doesn't work in everybody, but it seems to work in about 50% of the people. And we're still trying to understand the mechanism of why that is. So you can see more at tremorstop.com. So let's do some clinical updates. Now, there's got to be a separation of church and state here. We have a nutritional supplement that can handle many conditions that can improve your patients, like fatigue and glucose stability and endurance. These are important things, but we're also looking at potential drug applications. And so I don't want to confuse you here. A lot of these are not available yet and won't be available until the FDA gives us the nod. So there's current trials going on in cancer, stroke, Alzheimer's, Parkinson's, epilepsy, diabetes. It's a pretty extensive list, and it's because oxaloacetate is improving mitochondrial functioning. And mitochondrial dysfunction is tied to so many chronic diseases. It's the mantra, right? Again, we see that calorie restriction stops, or doesn't stop, but decreases cancer risk, decreases cancer metastasis. There's a study showing that in human lung cancer cells, it basically drove the cancer cells into senescence without hurting the other cells. And this is work by Angela Rubin, who's out of Israel. She looked at giving oxaloacetate to mice that were implanted with human glioblastoma tissues, saw basically a 50% reduction in tumor growth. When they combined the oxaloacetate with temozolomide, which is a chemotherapy agent. Here's the control. You can see this mouth is toast. Here's with oxaloacetate and chemotherapy. Pretty darn effective. Pretty darn effective. As a matter of fact, oxaloacetate alone performed in a similar fashion to chemotherapy alone, but without any side effects. When you combine them together, there was a 237% increase in survival, and 30% of the mice, they couldn't find the cancer tissue. Is that important? Oh, perhaps. FDA thought it was important. They gave us an orphan drug designation for glial tumors. And based on that, we went out and created a medical food. So this is now not a nutritional supplement, it's a medical food. It's a unique category set up by the Orphan Drug Act to help people with unfortunate diseases. And it allows us to immediately implement this into the marketplace. It's got to be made of food additives or GRAS ingredients. It's got to be used under a physician's supervision. That's not the same as getting a prescription, okay? It just means that a physician, and if you have a brain tumor, you probably have some physician supervision. And it must have a scientific basis. Well, obviously FDA thinks there is one because they gave us an orphan drug designation. And when we tried this in patients with a variety of glial-based tumors, such as astrocytoma and glioblastoma, 88% of them showed either the compound stopped the tumor or reduced tumor size. This is in brain cancer. It is not a cure, okay? It just stopped growth. Some of the tumors did continue to grow, however, so we're still working on it. And here are some of the case studies. Some of the interesting points, this was not done in a vacuum. The oxaloacetate, the Gliaxal medical food, was added to everything else we could throw at these tumors. So it included chemotherapy, radiation, surgery, everything we could possibly do, but through 88% of the people that we tried, it stopped tumor growth, at least for a while. Our data is very preliminary. You can see that the longest person here was 9 months. They had an anaplastic astrocytoma. They only did ketogenic diet after surgery and the Gliaxal medical food, and they saw a tumor reduction. So this is a good thing, and hopefully the data continues to pan out. Here's a quote from a doctor that his child is, got an anaplastic astrocytoma, 24 years old. He's doing a ketogenic diet, no chemo, no radiation. His prognosis is stable, and actually the tumor is getting a little bit smaller. Are we changing people's lives? Okay, so now we're starting a phase 1 clinical trial in pediatric brain cancer at the Rady Children's Center in Rady Children's Hospital in San Diego. They'll hopefully start this summer. And we're continuing case studies at UCSD, at the Moore Cancer Center, and at George Washington University in Washington, D.C. Let's talk about Parkinson's. I had a university professor call me up and say, I need a bunch of Benadryl. I was like, okay, sure, write a check. I said, now why do you need all this? Are you studying mice or what's going on? And she said, no, I have Parkinson's disease. And I said, okay, why do you want it? And she said, well, my neurologist suggested it for mitochondrial functioning. Okay. I said, have you tried it? And she goes, yeah, I tried it, and that's why I want to buy a bunch more. I said, is it working? She goes, yeah. So I arranged to have a PDQ-39, which is a validated survey, to look at before and after. And the higher the results of the survey, the more Parkinson's has affected her life. And you can see the red lines are before, and the purple lines are after. This woman was in a wheelchair. She couldn't stand for more than 2.3 seconds. She couldn't button buttons. She had trouble combing her hair. She had trouble feeding herself. But the thing that bothered her the most was she couldn't type on the keyboard. And she was a professor of pharmacology, and she couldn't type up her research papers. It's amazing what bothers people the most. And she started taking this, and as you can see, she's had a 90% reduction in the symptoms of Parkinson's. And this was 3 years ago. I talked to her last month. I said, how you doing? She goes, I still continue doing well. I said, how much Benadryl are you taking? She goes, well, I'm up to 3 capsules a day. And I said, okay, so it's going well. And she goes, yeah, I started jogging again. I'm like, she started jogging? She was in a wheelchair. She'd fall over. Are we changing people's lives? So we looked at a group of 12 people with Parkinson's, and what we found is that it seemed to be effective in about 50% of them. 50% had little or no improvement after 75 days. The rest had a partial response at 20 to 70% improved. The others had 70 to 100% improved. The average improvement was 65%, and it was statistically significant. As a result of this, we have gone into a clinical trial at the University of Kansas, imagine that. And it's a phase 2 double-blinded placebo-controlled trial. It's taken a little bit longer than I thought it would, but I should have some results in about 4 or 5 months. So we'll see. We also, did I get that one? We also looked at Alzheimer's disease. We got a large grant from the Alzheimer's Association, and this is work that was done at Louisiana State University. And what they looked at is putting oxaloacetate in the chow of the animals who have had their genes spliced with human genes to increase the amount of beta amyloid production. So they're a model for Alzheimer's disease. And here you can see that the secreted, here's the control, this is secreted APP alpha, which is considered a protective chemical, and this is every other day feeding, which would be equivalent to calorie restriction. This is maniheptalose, which is another calorie restriction mimetic that's not on the market yet, and this is oxaloacetate. So you can see they all had a protective effect, and it dropped intracellular A-beta 140 and A-beta 142, and dropped hippocampal plaque density, which is pretty cool, about the same as every other day feeding. And that plaque density decrease correlated with a decrease in short-term memory, or a decrease in the loss of short-term memory. So they gained about 50% short-term memory over the test animals that had this high amount of amyloid proteins in their brain. So it's not 100%, but we're well on the way of getting there. A phase 1 clinical trial is starting also at the University of Kansas. So obviously they like the initial results so far. So who can benefit from this? Well, right now we can only have you benefit as a nutritional supplement. So an obvious one is anti-aging. I mean, anything that's going to increase lifespans in multiple species has got a good shot of increasing human lifespan. We're not that different in animal. Help to maintain proper glucose functioning. There have been studies in the last 3 years that have taken biopsies of the pancreas, and instead of looking at gene expression, they look at protein activity. Okay, it's called proteomics. And one of the proteins in type 2 diabetics that they found are downregulated by a huge factor is pyruvate carboxylase. Its functioning is down as much as 65% as compared to normal controls that don't have type 2 diabetes. Now, in animal models, when we overexpress pyruvate carboxylase, the animals can't go into diabetes. That's interesting. And in animal models, again, when we disable pyruvate carboxylase in the pancreas, they almost immediately go into diabetes. Now, the function of pyruvate carboxylase in the body is to take pyruvate, act as an enzyme to break it into oxaloacetate. So essentially, these animals are going into diabetes because there is a shortage of oxaloacetate. So diabetes could be viewed as a shortage of diabetes, or as a shortage of oxaloacetate. Now, that said, we don't see 100% success in clinical trials. I mean, there was a clinical trial with oxaloacetate in diabetes. It showed 100% clinical success. But in talking to my doctors, I'm not hearing that. I'm hearing about a 50%. And I think a lot of that is because diabetes is a symptom of low blood sugar or high blood sugar, a problem with blood sugar. And there are probably many different causes for that. And if the cause is that your pyruvate carboxylase activity is down, then this is probably the best thing for you. But if it's a different cause, it may not have any effect on you whatsoever. So I'm not suggesting you use this for diabetes, but certainly it helps to keep normal glucose levels in healthy people. It helps support that whole cycle. Oxaloacetate is required because with oxaloacetate, when we give it to animals in their pancreas, it proliferates the beta cells in the pancreas. It helps to rebuild those cells that burn out so that you can have enough insulin to function. So it's an important nutrient. We also looked at improving mitochondrial function and density. It turns out that increasing the NAD to NADH ratio and activating AMPK is key to mitochondrial biogenesis. So we're actually able to build more mitochondria. So, hey, maybe you've got some sick mitochondria. Hey, we'll build more. What the heck, right? And this is one of the things that has been shown to do it, and perhaps is one of the reasons why it may be effective in Parkinson's disease, if that's what they find in the clinical trial. We've also seen it for weight maintenance. You know, it's not great as a way to lose weight, okay? What it does is it regulates the genes that create and store fat. And if you think about it, if your cell thinks that you're calorie restricted, you're not going to store fat. You're going to spend your time doing other things. And that's what we see. We see a downregulation of the genes that create and store fat by about 30%. And what that means is when you go on a diet, you have to do the hard work yourself. You have to lose that weight. But typically when you hit that spot, you're like, yes, I'm going to go get that ice cream sundae now. And you immediately rebound back, sometimes higher than your initial weight. And there are biochemical reasons for that. Your genes upregulate your ability to create and store fat because, hey, you were just in a famine. You need to store that because who knows when Piggly Wiggly is going to close again. Okay? So one of the things that oxaloacetate could possibly be used for is an after-diet supplement. You lose all that weight, and it helps you slow down that rebound effect. You saw the effects in the mice where there was a differential in the ability to gain fat. And that makes your diet more successful. That makes your clients happier with you. That makes your clients healthier for longer. You'll still gain weight. You'll just take a lot longer to gain it. The increasing endurance, fighting fatigue. You know, I've seen elderly people, you know, they shuffle, they get a little bit of oxaloacetate in their system. After about a month or so, they're walking into the office. It's a good thing. It's a good thing. You don't need to be an Olympic athlete to take advantage of an increase in endurance. Mood swings, this is a weird one. Mood swings within PMS seem to be tied to an increased spike in glutamate production in the brain. And one of the things we're looking at with oxaloacetate is it combines with serum glutamate through serum glutamate oxaloacetate transaminase, or SGOT, into alpha-ketoglutarate and aspartate. And in doing that reaction, it scours glutamate from the body and lowers glutamate levels in the brain. So we took a group of 12 women with severe mood swings in PMS, and out of that 12, 11 of them saw significant improvements to the point where they didn't have to go home from work 2 days a week. They could stay at work and actually be productive. I mean, this is changing people's lives. Constipation reduction. It turns out that oxaloacetate is one of the metabolic drivers of the lower intestine. So we see a lot of people who have constipation issues, it seems to go away. My mother-in-law, as a matter of fact, she doesn't take it for anti-aging. She feels she's lived long enough, but she takes it because it helps her every morning. And lastly, glutamate reduction. They're doing a lot of work in closed head injury, in stroke, in epileptic seizure, because it turns out that any kind of brain insult produces this glutamate bloom to protect the brain, presumably. But unfortunately, the glutamate is so active that it overexcites the neurons, and it leads to excitotoxicity. The neurons fire so much that they die. And this is one of the key culprits in brain cancer. The tumors excrete tremendous amounts of glutamate, and oxaloacetate can be used to reduce that in animal models of closed head injury. If they can get oxaloacetate to the animals within 2 hours, it reduces ischemia by 80%. Think about that. That's going into a clinical trial in Europe, and it will be exciting for the National Football League if it all turns out. As an experimental clinical drug, and again, this is not ready for prime time, we're looking at Parkinson's disease, glioblastoma, although we do have a medical food now for glioblastoma. Alzheimer's disease, H2-hydroxyglutarate aciduria, which is a rare genetic disease. Mitochondrial diseases, we're currently treating infants for a variety of these. Diabetes, these are all potentials. So for a clinical trial, we need IRB approval, data sent to clinicaltrials.gov, and you can follow along with us as we progress through this process. A patient approval form and 30-day FDA notification prior to beginning the trial. So I hope you join us in helping your patients with their issues with mitochondrial dysfunction, with fatigue, with glucose stabilization, with mood swings. Thank you very much. Short and sweet.
Thank you, Alan. Amazing. How do you keep track of all those studies? I've never heard of anybody doing so many studies. It's amazing. Clinically speaking, to get some of these results, what do we might need to do in dosage?
There are two types of effect with oxaloacetate. One is a genomic effect, and you've got to wait about 30 to 45 days to see it, because even though the genes are turned on and producing proteins that we want to see, it takes a while for the proteins to increase in density enough for you to see measurable effects. For glucose stability, we found that 2 to 3 caps per day spread throughout the day is pretty effective in about half the patients. The other half, it just doesn't seem to do a darn thing. So far in Parkinson's, we're testing 2 caps a day. We'll see if that's effective. In mood swings, it can be as little as 1 cap a day. Anti-aging, 1 cap a day. It's just important that you keep doing it every day for the genomic effect, because you want to keep raising that NAD to NADH ratio so that you keep the activation of AMP at a chronic level. The other effect is a pharmacological effect where you're trying to reduce glutamate through scavenging by oxaloacetate. And in that case, you have to really increase the dose of oxaloacetate quite a bit. Some of our glioblastoma patients are taking up to 60 capsules a day and have been for 9 months. You know, and looking at their blood levels, their blood levels are all stable, their liver enzymes are all stable. It's, you know, this is a human metabolite. The most noticeable thing is that they're still alive.
Okay, how about serum? Do we get serum levels on oxaloacetate?
I'm doing a study at University of Kansas that started actually yesterday. And as part of the phase 1 study for Alzheimer's disease, we're looking at serum levels both through oral dosage and then a sublingual dosage that we're developing.